Hello everyone, welcome to week 8 of lab, muscle contraction. I apologize about canceling lab this week. However, we are extending those due dates for the lab quiz and the lab report. So the muscle contraction lab quiz for you guys is going to be due on Friday, March 12th. So instead of this Sunday, it's going to be on Friday at 11:59 p.m. And the same for your lab report. So instead of submitting it before lab, you have an extension until Friday at 11:59 p.m. to get your lab report for full points. So even though lab is coming at you later, you're going to have some time to still work on the lab report and the quiz. Okay, so muscle contraction. This is excitation-contraction coupling and the sliding filament mechanism. We bring those two things together, and that results in muscle contraction. Here's our schedule. I'll lecture on the neuromuscular junction and the sarcomere. We're going to make sense of excitation-contraction coupling and the sliding filament mechanism. We're going to do a quick worksheet of the muscle contraction steps that I emailed to you. It's not on Blackboard. It'll be found in the email that the link to this video is in. We'll go over the procedure. So we're watching a couple of YouTube videos, We'll talk about the lab report, and then we'll talk about what's to come next week. I personally like to kind of visualize where in the nervous system I'm working whenever I talk about the nervous system, because it can kind of get tricky. So where are we right now? We're in the peripheral nervous system, which has an afferent division, so going towards the central nervous system, and an efferent division going away from the nervous system. If we're talking about the neuromuscular junction, we're talking about the junction between the neuron and the muscle. So this is going to be the somatic division of the efferent part of the peripheral nervous system. So we see that output from the CNS can be either coming from the somatic nervous system or the autonomic nervous system. When we're talking about the neuromuscular junction, we're talking about the somatic nervous system, which is working on motor neurons, which the motor neuron is then going to work on a skeletal muscle. Here's some purposes for us today. We'll describe the structure and function of the neuromuscular junction. So kind of a review. We will apply knowledge of synaptic physiology to relevant clinical scenarios. We will explore the basic structure of the sarcomere, and we will learn about those contractile, regulatory, and structural proteins. And finally, we will describe the events of excitation-contraction coupling and the sliding filament mechanism to bring together the big picture of muscle contraction. First off, this is actually part of the procedure to watch this video. So this details motor units and the events at the neuromuscular junction. So this should be a review to you. Hint, one of the questions to your lab report is found in this video. Let's jump right in.
The body movements are controlled by muscles. While contracting, the muscle is reduced in length and joins closer the two bones on which it's inserted. However, it's the nervous system that controls the muscle contraction by the means of nerves. Each nerve contains thousands of nerve fibers arranged in dendrites, which carry out sensory information, and axons that convey motor impulses. Each motor neuron innervates several muscle fibers. This association defines what is called a motor unit. In general, the fewer the muscle fibers are in a motor unit, the more the movement is precise. For example, in the temporalis muscle, there are 1,000 muscle fibers per motor unit, while in the external ocular muscle, there are only 5. This reflects the accuracy of the eye movements.
So that was part of the answer to one of the questions on your lab report. Not the full answer, though. We need to relate that to the precentral gyrus, right? Okay,
The
back to
intensity
skeletal
of the muscle contraction is proportional to the number of motor units recruited. A single motor neuron gives several endings that sometimes scatter throughout the thickness of a muscle. Each ending is intended to stimulate a single muscle fiber in a specific place, the neuromuscular junction. Just before the axon terminal, the neuron loses its myelin sheath and forms a terminal button.
Terminal button, also known as the synaptic end bulb.
The terminal button contains many mitochondria that provide energy and several synaptic vesicles. Each vesicle contains approximately 10,000 of acetylcholine molecules, the unique neurotransmitter of the neuromuscular junction. On the side of the muscle fiber, we find the motor end plate, which is the area directly opposite to the terminal button. The motor end plate, thick and electrically non-excitable, forms folds that increase the volume of the synaptic cleft. Although these two regions are very close to each other, there is no real contact between them. Once it reaches the nerve terminal, the motor impulse causes the opening of calcium channels, triggering an influx of calcium ions inside the cell.
So we're still on the synaptic end bulb here. These are the voltage-gated calcium channels opening up, allowing calcium to rush in.
The calcium ions promote the fusion of acetylcholine vesicles with the cell membrane, releasing their full content of this neurotransmitter in the synaptic cleft. The acetylcholine molecules diffuse then towards the other side to cholinergic receptors accumulated mainly in the folds.
Cholinergic receptors are those neurotransmitter receptors that are on this postsynaptic cleft.
The binding of two acetylcholine molecules to one receptor causes the opening of a sodium channel that facilitates the entry of sodium ions in the interior of the muscle fiber. This depolarizes the membrane and creates motion.
I'm just going to pause here because it said influx of sodium ions. What do you know about that? When sodium ions come rushing into a cell, that means that more positive charges are rushing into a cell, causing resting membrane potential to become closer and closer to zero, making it more excitable, more likely to start in action potential. So that's what he's saying when this depolarizes the membrane.
And plate potential. Depending on the number of activated receptors, that potential may exceed a threshold and trigger a muscle action potential, which will broadcast to the entire muscle membrane and cause a contraction of the muscle fiber.
So action potential propagated along that axon, release neurotransmitter, causes another action potential to spread along the muscle fiber, which causes contraction.
The acetylcholine molecules are rapidly destroyed by an enzyme, acetylcholinesterase, present in the synaptic cleft. This lysis will give two molecules of acetate and choline, which joins the nerve endings to help form new molecules of acetylcholine.
You know about that step. That's one of the fates of the neurotransmitter. Acetylcholine esterase can come in and degrade acetylcholine.
The rapid destruction of acetylcholine allows avoiding the prolongation of muscle contraction. There may be a small release of acetylcholine by spontaneous exocytosis in the synaptic cleft without any nerve stimulation. However, the number of activated receptors is far from triggering a muscle action potential. A note.
Okay, that was our kind of overview, review, intro thing of the neuromuscular junction. I made the slide about the neuromuscular junction, but we literally just talked about it. So simply just remember that the neuromuscular junction is the space where a motor nerve synapses with a muscle fiber. So it's the space where the motor nerve is communicating with the muscle fiber, translating the action potential from the motor nerve to that muscle fiber. And this is where excitation contraction coupling happens. So maybe pause this video right now and do a brief neuromuscular junction review. See if you can identify all of these things on the side here in this picture. So that was our neuromuscular junction. If we look below here, within the muscle fiber, we have these contractile elements. So you see kind of these striations right here. This is the muscle fiber. Those striations are actually sarcomeres. So this is what it looks like in real life if you were to look at it with a microscope. And then this is kind of the picture of it drawn out. You don't need to know the I band, H zone, A band, any of that stuff. You just need to know where the Z line is, which normally it's that like squiggly Z looking line. But anyway, the sarcomere is going to be the functional unit of a skeletal muscle fiber. So we know that a functional unit is the smallest unit that can perform the function of whatever thing we're talking about. So within muscle, the sarcomere is the smallest unit that can perform a muscle contraction. You see in this GIF here, that's a sarcomere shortening. So you have, that's a myofibril right here. Within the myofibril, there's a bunch of sarcomeres. And each individual sarcomere will shorten during a contraction, leading to the shortening of the entire muscle. In this picture here, you need to know about the Z line, the thin filaments, the thick filaments, and there is also a couple of proteins not pictured on this little diagram. But you need to know contractile, actin, and myosin. Those are going to be the thin and thick filaments. Regulatory proteins, troponin and tropomyosin, and then structural proteins. This is going to be titin. So those are the muscle proteins that you need to know that lie within a sarcomere. And here they are in a little bit more detail. I correlated the colors of the word with whatever color it looks like on here. Sorry if you're colorblind. But we will see that the contractile proteins are actin, which is the thin filament. So actin is going to be this red part right here. If you look at it a little bit closer, those are actin. And we describe actin as kind of a string of pearls, two strings of pearls wrapped around each other. And then myosin is going to be the thick filament right down here in this diagram. It's this purple. This is it closer up. And this is our thick filament. So all of these little branches popping up, if you look at it closer, has a myosin tail and a myosin head. The myosin head is going to connect to actin and move things along. Our regulatory proteins are both going to be on actin. So troponin, well, first, tropomyosin is this kind of string thread-like protein that lies along those actin active sites. So it covers the actin active sites. That's why it's a regulatory protein. And then troponin is going to be like a tack that sits on top of tropomyosin, keeping it in place. The way that I remember the order of troponin and tropomyosin, because they're both a T word that sound kind of the same, is that troponin is on tropomyosin. So troponin is on top of tropomyosin. That's how I remember it. And our structural protein is going to be titin, this green squiggly thing. This is going to be, this attaches myosin to the Z lines, which allows, if you have too strong of a contraction, it isn't going to like totally ruin your sarcomeres that titin is in there working as a molecular string, absorbing that shock. So now that we have reviewed the neuromuscular junction and we are familiar with a sarcomere and the proteins that make up a sarcomere, we can talk about excitation-contraction coupling and the sliding filament mechanism. Excitation-contraction coupling is defined as the rapid communication between electrical events occurring in the plasma membrane of skeletal muscle fibers and calcium released from the sarcoplasmic reticulum. So the electrical events that occur in the plasma membrane of a skeletal muscle fiber. That's what this GIF up here is showing. Action potential propagates along, sent down the T tubules, and those little balls that are released would be calcium. So again, action potential propagates along the sarcolemma, down the T tubules, and calcium is released. That's all electrical. That electrical stuff leads to mechanical contraction. When I think about electricity, I think about like blues and greens, and when I think about mechanical contraction, I think about red because muscle. So I put the electrical events there in green for you, and then mechanical contraction in red. So this excitation of the muscle fiber leads to contraction via the sliding filament mechanism. What is the sliding filament mechanism? This is simply the concept that muscle proteins slide past each other to generate movement. Really, it's, myosin attaching to actin, and actin is sliding over myosin. So we would say that actin slides over myosin, but the filaments are sliding to cause muscle contraction after being electrically stimulated. Excitation, contraction, coupling. I know I'm being redundant right now, but I would rather be redundant than not specific enough, I guess. Here are our muscle contraction steps. So now that we know about excitation, contraction, coupling, and the sliding filament mechanism, we could put those two things together to talk about muscle contraction as a whole. So let's jump right into it. Here are our steps. So the action potential travels down the motor neuron to the neuromuscular junction, and that is synaptic transmission. That action potential travels down the T tubules. So once the action potential travels down the motor neuron to the neuromuscular junction, synaptic transmission occurs, right? The transmission of the action potential coming from the motor nerve is transferred to the muscle fiber, if you will, and that action potential is going to propagate along the sarcolemma, again, in this GIF, and then now we're at step 2, action potential travels down the T tubules and calcium is released from the sarcoplasmic reticulum. I put a little bar here indicating that these first 3 steps up until calcium being released from the SR, that's electrical. So electricity, I guess you could say, is what's stimulating all of that to happen. Action potentials is what's stimulating all of that to happen. So action potentials stimulates calcium to be released from the sarcoplasmic reticulum. And then at number 4, we start the mechanical part. So once calcium is released from the sarcoplasmic reticulum because it was stimulated to by the action potential that traveled down the T tubules, that calcium is going to bind with troponin, which is on top of tropomyosin. So once calcium binds to troponin, it allows tropomyosin to shift, which exposes actin active sites, as you can see in this schematic down here. Once those actin active sites are exposed, we learned that myosin heads have a very strong affinity for actin active sites, which is why we need troponin and tropomyosin on those actin active sites when we don't want to do a muscle contraction. So once calcium is there, exposing those actin active sites because it bound to troponin and tropomyosin, myosin is immediately going to form a cross bridge with actin, just simply because it's exposed now. So that's number 6, and that's this picture right here. This is the cross bridge formation. Once the myosin head makes contact with the actin active site, this is what we call the cross bridge. From there, once it attaches to the actin active site, that ADP, adenosine diphosphate, and inorganic phosphate are going to be released from the myosin head. And the release of the ADP and PI, inorganic phosphate, is going to release energy, which causes that power stroke. So this is kind of like the motion that we use when we're describing how a myosin head moves. It's moving the actin along. So it attaches with the actin. That's the cross bridge formation. ADP and PI are still there, but once it attaches, ADP and PI are going to be released, and that releases energy for the myosin head to move that actin strand along and allowing the sarcomere to shorten. So the myosin head bends, power stroke, actin slides over myosin, steps 8 and 9. From there, once the power stroke occurs, a new ATP molecule is going to come along and bind to myosin, to detach it from actin. And what do we know about this myosin head? What enzyme is on here? I, for some reason, can't think of the enzyme right now, but you know it. And it's going to split an ATP molecule into ADP and PI. So what's going to happen right when a new ATP molecule comes along, detaches the myosin head from the actin, right? Once that happens, ATP is immediately going to be converted into ADP and PI, and unless there is more calcium there to bind to troponin and expose the actin active sites, because troponin is not tropomyosin, tropomyosin shifts. As long as there's more calcium there exposing those active sites, that myosin head is going to go back up to step A-B and make that cross bridge formation. But if those actin active sites are not exposed, our contraction is done. So muscle contraction is another one of those things like osmosis, where you learn it and you're like, oh, that makes perfect sense. And then you go to answer a question about one of the steps in muscle contraction and you're like, wait, where does it begin? Where does it end? It's kind of like a never-ending circle. So spend some time with this. Muscle contraction is, those of you who are in the exercise science major, muscle contraction is literally like the holy Bible of what you need to know in that major. So definitely spend some time with this, make it make sense. I have some videos to show you. This is again part of our procedure, but I think these videos will kind of bring it all together and help you visualize what it actually looks like. So this video is going to help you visualize the events of muscle contraction.
Stimulates a muscle cell. An action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads from the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction. In each contracting unit, or sarcomere, thin actin and thick myosin filaments are juxtaposed, but cannot interact in the absence of calcium. This is because myosin binding sites on the actin filaments are all covered by a rod-shaped protein called tropomyosin. A calcium-sensitive complex, called troponin, is attached to the end of each tropomyosin molecule. When calcium floods the cell, troponin binds to it, moving tropomyosin off the myosin binding sites. Opening the myosin binding site on the actin filaments allows the myosin motors to crawl along the actin, resulting in a contraction of the muscle fiber.
So there you see the actin is in red, myosin is in green. So this thick filament is myosin, the thin filament is actin. And this shows how actin is being pulled towards the middle by those myosin heads. So the myosin heads are like going like this, and then the actin is sliding along. I mean, you see in the video.
Calcium is then quickly returned to the sarcoplasmic reticulum by the action of a calcium pump. Without calcium, myosin releases actin, and the filaments slide back to their original positions.
So that was that, and here is one final short video. This shows a close-up of the interaction between myosin and actin, so really close up. And it describes how ATP is hydrolyzed to drive the power stroke. So it goes a little bit more in depth for those of you who like to make more sense of things when things are explained in grave detail.
Striated muscle is made up of bundles of multinucleated muscle cells called myofibers. A myofiber is packed with myofibrils that extend the length of the cell. A sarcomere, the functional instructional unit of muscle, consists of thick myosin filaments and thin actin filaments. Tightening keeps thick filaments centered during contraction. In muscle contraction, actin filaments slide past myosin filaments. Neither filaments change in length, yet the sarcomere becomes shorter.
I really like the way that they visualize this schematic, how it's a circular thing, showing that the sarcomeres Z to Z aren't this just like box-like thing that we see a picture of. It's a multi-dimensional, kind of like, looks like a wheel if you were to turn this picture and look at it. It's really hard to do this virtually, but you get what I'm trying to say. It's cool.
The force required for this movement is generated by myosin heads, which make cross-bridge attachments to actin. Myosin heads walk along an actin filament, using ATP for energy.
Myosin is pink, actin is green.
Is part of the complex that prevents myosin from sliding along the thin filaments.
This is just upside down of what you're used to seeing. Normally you see myosin below actin, just like in the diagrams that I've shown you so far. This is the same thing, just upside down.
A myosin head binds ATP, and as a result, detaches from the actin filament. It rebinds in a new position. Phosphate release triggers a power stroke that moves the actin filament.
So that was the inorganic phosphate being released, causing the power stroke.
ADP disassociates, and the process is repeated.
So the steps are intricate, but this is a really quick thing. I mean, even this schematic is moving really slow. But imagine that happening like really super duper fast in every single sarcomere in your myofibril that's being contracted. Now, at this point in lab, we would have done a breakout session to go over the muscle contraction step exercise. That document can be found in the email that I sent to you that has the link to this video that you're watching right now. So this exercise is not on Blackboard, it's in your email. So just going to go over this quickly. Here's our exercise. I don't feel the need to read over all of this. You guys can fill that in. You know the answers. Here's your word bank. You have too many options here, so you aren't obviously going to use all these, but all the correct answers are in there. Notice this asterisk at the bottom. Draw a line where excitation contraction coupling stops and the sliding filament mechanism begins. So this is where electrical precedes mechanical. So I'm going to give you the answer here. The difference between electrical and mechanical is found in calcium release and calcium binding. So we know the last part of the electrical part of excitation, contraction, coupling is going to be calcium release from the sarcoplasmic reticulum because an action potential was propagated down those T tubules to allow calcium to be released from the sarcoplasmic reticulum. So this calcium is released from the, just gave the answer. So that's going to be the end of electrical. Whereas right here, when calcium then binds with blank, this is going to be the beginning of mechanical. So I know that you have done this worksheet, or excuse me, I'm being distracted by outside noise. I know that you've watched this video if you completed this worksheet and showed me where that difference is. So if you were to draw a line, it would just be between here, right? Okay, so go ahead and attach that muscle contraction exercise to the end of your lab report. It'll be like a participation points kind of thing. And now we're going to tackle these applications quickly. So in your lab report, you have 3 exercises to do. I think it's part 4 applications, and then there's 3 exercises. Excuse me. Please go ahead and fill this out, answer it, and attach it to your lab report at the very end. I'm going to go over the answers here. Our first exercise is Lambert-Eaton syndrome, LES. This is an autoimmune condition. We know that autoimmune diseases are diseases where the body's own immune system attacks its own cells, autoimmune. In the case of LES, antibodies are inappropriately targeted against voltage-gated calcium channels on the synaptic end bulb of the neuron. This prevents calcium from entering the synaptic end bulb and triggering the fusion of acetylcholine vesicles with the synaptic membrane for release. A couple of questions to answer here. Number 1, what would be the symptoms of LES? If we think about our neuromuscular junction, so we know that LES antibodies are inappropriately targeted against those voltage-gated calcium channels on the synaptic end bulb of a neuron. So if calcium isn't able to come into the neuron, I have drawn circles and X's around these voltage-gated calcium channels. If calcium can't come in because those voltage-gated calcium channels aren't working, then acetylcholine within the vesicles isn't going to be released from the neurotransmitter. And if acetylcholine from the motor nerve. And if acetylcholine isn't released, then we won't have a contraction at the very end. So if we don't have a contraction at the very end, some symptoms of LES would be weakness, difficulty contracting your muscles because you're having issues with those voltage-gated calcium channels. Number 2, besides affecting the somatic nervous system, people with LES also experience symptoms related to the dysfunction of the autonomic nervous system, although these symptoms are relatively mild. Use what you know about the autonomic nervous system to determine why this disease is also causing problems in the ANS. What's our mechanism here? In the case of LES, antibodies are inappropriately targeted against voltage-gated calcium channels on the synaptic envelope of the neuron. This prevents calcium from entering the synaptic envelope and triggering the fusion of acetylcholine vesicles with the synaptic membrane for release. What do we know about the autonomic nervous system? We know that within the autonomic nervous system, we have a parasympathetic branch and the sympathetic branch. Parasympathetic is going to be that long presynaptic neuron, short postsynaptic neuron. Sympathetic, short presynaptic neuron, long postsynaptic neuron. So what neurotransmitters are we working with here? In the parasympathetic nervous system, the way that I remember this is that you're working with a pair of acetylcholines. So we're going to see that the neurotransmitter used at both the pre and post synaptic neuron is going to be acetylcholine. What about sympathetic? Neurotransmitter released from the presynaptic neuron. Again, acetylcholine here. What about the postsynaptic neuron of the sympathetic nervous system? There is going to be norepinephrine or epinephrine released here as a neurotransmitter. So if we know that with our case of LES, acetylcholine isn't going to be released because those voltage-gated calcium channels aren't allowing calcium to rush in to signal acetylcholine to be released. So we were asked if this exerts its effects on just the somatic nervous system, the neuromuscular junction, or if it can also exert effects in the autonomic nervous system. Looking at our neurotransmitters that are being used, the answer is yes, it can also affect the autonomic nervous system because it's going to be working on wherever acetylcholine is being released. So in the case of LES, we would see that there's difficulty releasing acetylcholine both from the presynaptic neuron and the postsynaptic neuron in the parasympathetic nervous system. And in the sympathetic nervous system, there is going to be difficulty releasing acetylcholine in the presynaptic neuron, which if you have difficulty communicating that to the postsynaptic neuron, it's probably less likely to release that norepinephrine or epinephrine, right? So that was our first exercise. Exercise number 2 is about nerve gas. We know that there are some chemicals, such as nerve gas and also commonly used insecticides, that cause irreversible inhibition of acetylcholine esterase, ACHE. Whenever we see ACE, we know that we're working with an enzyme that catalyzes reactions. So acetylcholine esterase is the enzyme that's responsible for degrading acetylcholine into choline and acetic acid in the synaptic cleft. That's one of the fates of the neurotransmitter in synaptic transmission. This results in the accumulation of acetylcholine throughout the nervous system, resulting in overstimulation of both muscarinic and nicotinic receptors. And atropine can be used to combat the effects of this. We have a few questions here. What effect would an acetylcholine esterase inhibitor have on the events of the neuromuscular junction? Think about that for a second. If we have an acetylcholine esterase inhibitor, so something that's inhibiting the action of acetylcholine esterase being the enzyme that breaks down our acetylcholine. If we have an acetylcholine esterase inhibitor, what effect would this have on the neuromuscular junction? If we aren't able to come in and get rid of that acetylcholine, then the acetylcholine is going to continue to bind with receptors on the postsynaptic membrane, right, on the on the muscle fiber. And as long as acetylcholine is still binding to those receptors, contraction is going to keep on happening. And so if an acetylcholine esterase inhibitor is there inhibiting the activity of acetylcholine esterase, we'll see that accumulation of acetylcholine causing a constant contraction, or we refer to this as tetany. Number 2, would acetylcholine esterase inhibitors have an effect on the somatic or autonomic nervous system, or both? We know that within the somatic nervous system, we're talking about muscles, so the skeletal muscles, voluntary movement of skeletal muscles, and So with the somatic nervous system, voluntary movement of skeletal muscles, neuromuscular junction. At that neuromuscular junction, the neurotransmitter that we have been dealing with, is acetylcholine. So we know it's definitely going to affect the somatic nervous system. But what about the autonomic nervous system? Well, refer back to the picture that I just drew, and also in this little schematic on the bottom. And we just learned that in the parasympathetic, there's a pair of acetylcholine neurotransmitter molecules being released both from the pre-ganglionic fiber and the post-ganglionic fiber. And then in the sympathetic nervous system, acetylcholine molecules are being released from only the pre-ganglionic fiber. So we would see, yes, acetylcholine esterase inhibitors will have an effect anywhere where there is acetylcholine and acetylcholine esterase. So yes, indeed, we will see that ACHE inhibitors work both on the somatic nervous system and the autonomic nervous system. The autonomic nervous system branching off into the parasympathetic and sympathetic. Within the parasympathetic, there's a pair of acetylcholines being released from both the pre and the post. Sympathetic acetylcholine is only being released from the presynaptic neuron. Okay. Number 3, atropine, which is given in an auto-injector, is the standard treatment issued to soldiers to combat nerve gas poisoning. So this acts as an antagonist or a blocker for muscarinic acetylcholine receptors. With that information, would this treatment completely reverse the effects of nerve gas? So in order to answer that, we need to know the difference between muscarinic and nicotinic receptors and adrenergic in all of those. So if you look at this little schematic on the bottom, in both the actually I'm going to draw it out. Here is our autonomic nervous system, parasympathetic and sympathetic. So acetylcholine being released in the parasympathetic as well as in the sympathetic from the preganglionic fiber. what are these receptors going to be on the postganglionic fiber? What have you learned? These are nicotinic. So not going to work there, but it will work here because on this effector organ, these receptors are muscarinic. All right, so these are muscarinic receptors. They receive acetylcholine, whereas these receptors at the postsynaptic neuron are going to be nicotinic receptors, still receiving acetylcholine, just a different type of receptor. So if we know that within the autonomic nervous system, by the way, this is down here, this is adrenergic. In case you just want the whole picture, these receptors here are going to be adrenergic. So we know within the autonomic nervous system, the parasympathetic division would be the only one that's affected because that's the only one that has those muscarinic receptors, right? So the question is, would this treatment completely reverse the effects of nerve gas? Not completely, because we were only working on those muscarinic receptors, which are only at the bottom of the parasympathetic branch of the autonomic nervous system. So if we had something that maybe worked on nicotinic receptors too and adrenergic, that'd be great. Our final exercise is about botulism. So this is a potentially fatal paralytic condition caused by Clostridium botulinum. That is a bacteria, anaerobic gram-positive spore-forming bacterium for you microbiome weirdos. The bacteria produces a toxin that prevents neurotransmitter vesicle anchorage and acetylcholine release in the synaptic cleft, preventing muscle contraction and resulting in flaccid paralysis. This condition, botulism, most commonly occurs due to the ingestion of botulism-containing food. Common culprits for this are improperly canned food. So in the infant, the ingestion of raw honey contaminated with spores of this bacteria leads to paralysis, described as floppy baby syndrome. So if babies get this, it can not be good because when you pick them up, if you look up pictures of this, it literally looks like you're holding up like a puppy instead of a baby. You know, when you hold up, I don't know if you've ever held up a puppy, but they just kind of like lay there because they're sleeping and they're so floppy. That's what it looks like in floppy baby syndrome, and I don't mean to laugh because it's actually a serious thing. But normally when you pick up a baby, they should be like wiggling around and all that, but in floppy baby syndrome, pick up the baby and it's just flaccid. Excuse me. How this paralysis begins in the muscles supplied by cranial nerves of the face. So this causes blurred vision, trouble speaking, swallowing. And then unless you interrupt it with an antitoxin to get rid of this toxin, this will eventually progress to the respiratory muscles, making it difficult to breathe, impossible to breathe without mechanical ventilation to the patient. Here are our questions. Draw the neuromuscular junction and indicate where the toxin exerts its effect. So go ahead and draw that out right now on a little piece of paper. Here's my drawing. So here's our synaptic end bulb. We have vesicles containing acetylcholine, our neurotransmitter. Here it said, bacteria produces a toxin that prevents neurotransmitter vesicle anchorage and acetylcholine release from the synaptic cleft. So prevents acetylcholine vesicle anchorage and release. So at this part right here, crossed out, that's where it's exerting its effect. Acetylcholine neurotransmitter isn't being released into that synaptic cleft. Number 2, would all of your cranial nerves be affected? Why or why not? We learned about the cranial nerves. Well, you learned about them back in anatomy, but we reviewed them a little bit ago. And I had you categorize them as either sensory, motor, or both. Since we're talking about the neuromuscular junction in the somatic nervous system, so voluntary motor movements, right? Only the nerves that fall under that motor or both category are going to be affected by botulism. So that's going to be your oculomotor, trochlear, trigeminal, abducens, facial, glossopharyngeal, vagus, accessory, hypoglossal. So no, not all cranial nerves would be affected, only the ones that fall into the motor or both category. So like I said, go ahead and answer the questions to these exercises that are in your lab report based on what I said in this video. Literally just write down what I said and include that with your lab report that you send to me for kind of participation points, quote unquote. Now let's take a look at your lab report. If you want to go back and watch the videos, I played all these videos today. Also, there's some really good information up here. Excitation, contraction, coupling, and the sliding filament mechanism. Professor Wallace wrote it all out right here in detail. So anyway, talking about the lab report, and I apologize if you hear hammering. I guess someone's doing construction downstairs right now. So questions 1 through 4. Hand draw. So I suppose maybe I'll accept if you have a tablet and one of those cool pen things and draw it that way, but you can't just like send me a diagram. Hand draw on a piece of paper a relaxed and a contracted sarcomere and label the following. So you're going to draw 2 different sarcomeres and you're going to label the Z lines, the actin, myosin, troponin, and tropomyosin. So here's our sarcomere right here, this thing. It doesn't need to be in this grave of detail, but for the difference between a relaxed muscle and a muscle that is contracted, what differences are we going to see in those drawings? So imagine maybe this is a relaxed muscle. If this were to be contracted, we would see that these Z lines right here, so where my pointer is right here and here, those Z lines are going to get closer together because those myosin heads are pulling actin in. So maybe one sarcomere that you draw is like this length, and then the next one that you draw is like this length. However, the length of the proteins doesn't change, just the degree of overlap is changing. So if you're still kind of struggling with that concept, go ahead and Google relaxed versus contracted sarcomere. I'm sure you'll find some good stuff on there. But please hand draw me 2 different sarcomeres, one relaxed, one contracted, labeling the Z lines, the actin, the myosin, troponin, and tropomyosin in each one. I'm sorry, Jenny. Number 5, describe how acetylcholine triggers an excitatory postsynaptic electrical signal action potential on the motor end plate. Go back to the events of synaptic transmission. Number 6, in your own words, so we know that, if you scroll up, you could literally just copy and paste all that, but don't do that. In your own words, describe the steps of muscle contraction. And then, so this is referring back to that worksheet, which steps above are describing excitation contraction coupling, and which are describing the sliding filament mechanism. So make sure you maybe draw a line or indicate where it goes from. This is the end of electrical stimulation, and this is where mechanical contraction begins. Number 7, in an experiment on synaptic transmission, a synapse between a neuron and a skeletal muscle fiber was kept in a solution equivalent to the extracellular fluid, but without the calcium ions. An electrical stimulation was applied to the neuron, but the usual response of the muscle did not occur. Considering the physiology of synapses, discuss what conclusion the researchers reached based on the result obtained. So what did these researchers just discover in the realm of muscle contraction? They said in this experiment, the synapse between a neuron and a skeletal muscle fiber was kept in a solution equivalent to extracellular fluid, but without calcium ions. So no calcium ions were there. They sparked that nerve with some electricity to initiate an action potential, but that action potential wasn't, nothing happened to the muscle fiber. What did these researchers discover? And number 8, last week we learned about the motor homunculus, in which we saw the precision of movement was related to the volume of cortex devoted to moving a body part. This week we learned about motor units and how the ratio of motor neuron to muscle fiber influences the accuracy and control of movement. Describe the precentral gyrus and the motor unit arrangement to justify the fine motor control we experience over the extrinsic eye muscles. So answer to that, refer maybe back to last week's lab, information on lab 2 weeks ago, about the motor homunculus, right, and the precentral gyrus, different cortical areas associated with your ability to carry out different movements, right? You answered that question on your lab report. So describe the precentral gyrus and the motor unit arrangement to justify fine control we experience over the extrinsic eye muscles. Go ahead and look at that first YouTube video again if you can't think of the answer there. Number 9, botulism injection. Botulinum toxin injection, which is Botox, for treatment of facial wrinkles is the most frequently performed cosmetic procedure in the United States. Based on your knowledge of the botulism toxin mechanism of action, explain how Botox acts in aesthetic treatments to reduce forehead wrinkles. So, what is happening there? Refer back to that exercise, the botulism exercise, to answer this question, but what's happening there to make wrinkles go away? And then finally, give me a citation, something that has to do with this topic, right? That's your lab report. Now, that's it. That's muscle contraction. What do you have to do between when you watch this video and lab next week? Technically Friday next week. Take that lab quiz by Friday the 12th at 11:59 p.m. and email me your muscle contraction lab report by Friday the 12th at 11:59 p.m. Again, giving you an extension just this one time because you're getting this video later than expected. So next week, we are going to be talking about muscle physiology in lecture. Next week, week 9, muscle physiology in lecture, and then for lab is going to be a Q&A for exam 2, which exam 2 is on March 15th. So that's coming up week 10. Those of you who were in my Thursday lab and in Khadijah's lab, you're good to go with that information. But people who are in my Wednesday lab, next week is a wellness day. So we will be doing something different for lab. I'll just communicate that with you. But just so you know, this upcoming Wednesday is a wellness day. So unfortunately, I won't be seeing you for 3 weeks in a row. So that's all the information that I have for you. I hope that muscle contraction is kind of starting to make sense, because this is a really big topic and something that is just really important for you to know, especially in the exercise science major. So as always, let me know if you have any questions about this. Reach out to other GAs, Khadijah students, reach out to her. Reach out to your SI, reach out to Professor Wallace if you have any questions. We want to help you, especially with this second exam coming up. The skeletal muscle stuff is going to be on there. It's going to be on there. Like, she's going to make sure that you know that stuff. So any questions, please reach out. Otherwise, I hope you have a good weekend, and I will talk to you soon.